It seems all encompassing, and I was wondering, how do I do this? It's a training. It's also like a continuous education program. I decided to just take a certain part of it to discuss. I wanted to discuss the relevance of an industrial pharmacy practice. A lot of things get lumped into industrial practice. Sales people, marketing, production, quality control. How do you drive clinical outcomes? The field of manufacturing practices, sales practices, importation and distribution practices, quality control. And I added at the end of it that we should look at basic clinical trial processes and the interpretation of clinical data. I don't know, I can ask you, what do you think? Marketing, sales, okay, and yet, sales, okay. So have you sell prescription drugs? So you know that means there are the clinical papers that have been published for the particular product you're selling. Have you ever been given any? Okay. So we'll not talk a bit about that, because sometimes now that is what distinguishes from another sales or another marketing person. Let's say you walk in the office, how do you feel that your drug is better? I have this clinical paper dropped off, and you say, I want to see it. If you see it, what do you look for? Let's discuss clinical outcome first. What is it? If you practice industrial pharmacy like we just talked about, there is a big disconnect between those who practice this and the patient. You don't see them, they don't see you. How do you know the drug is promoted for them or they didn't know? So the outcomes can be so varied from health to disease mitigation. Used to say to reduce morbidity and mortality when taking drugs. But outcomes in this day and age is way beyond that. It can be any measure of both the actual disease itself or surrogate, what we call surrogate outcomes. Surrogate outcomes are more like lab results. Okay, you give this person this, and the lab is no longer safe. But that may not translate to clinical, any good clinical outcome for the patient. So when you are doing these things, you make sure a lot of companies claim because the surrogate outcome performed so well, they will be hyping that. But it's not necessarily translating into clinical outcomes. So outcomes can be anything that is the goal of the therapy that you're doing or that the patient is taking. Medication, do you think there will be any credibility to medical practice if you don't have medicine? Not sure about that. And that is correct. It is true. If we don't have medication, there is no credibility for medical practice at all. But that is not to say there are other things in medical practice that does not need medication. You have radiology, surgeries, even though you say, okay, surgical patient will still take pain medicine or antibacterial, or even the anesthetic drugs that are given are drugs. The appropriate manufacturing, distribution, and sales is what will guarantee the outcome. And I can, when we go further, I will let you know individually where most people have a disconnect between clinical outcomes and any of these functions. Sales and marketing teams, particularly sales here, your responsibility is to communicate accurate medication use to prescribers. If you fumble that part of it, it's likely that prescriber may not use the drug appropriately. The importance of feedback in the whole of this practice is important. Okay, if you go back with a sales rep and you go to a doctor's office, it is good to ask if they prescribe the drug. Has any patient been back and telling you how your drug works? If you have that feedback, this is you trained concern for the outcome. If there is a problem with it, it's your responsibility to take it back to the company and say, this is what I heard. And there is actually where I come from, there is a very good reporting system for things like adverse events, unintended events, that they give you something for malaria and it's causing something. So if you hear that kind of feedback, you are obligated to take it back to the company. So that's the part that put the feedback. Good manufacturing practice. I've never been in production before, but it should guarantee consistency of drug delivery. A lot of people, I don't know about here, a lot of manufacturing companies have standardized procedure they use for manufacturing that guarantees whether it's a tablet, a capsule, or any of those, they have to deliver the same medication each time. It's a promise that you are making, both in bioavailability and bioequivalence. Even a small deviation from batch to batch can affect the patient outcome. Depending on, if you guys remember pharmacy school, the therapeutic index of the drug. I mean, if he has a very narrow therapeutic index, it's not as forgiving as something with a large therapeutic. They used to tell us that was digoxin, for instance. If you make a mistake, even a very narrow mistake in digoxin dosing, either due to your error or the error of manufacturing, they are likely to see a big difference in the outcome for the patient. Are you keeping the patient in view when you are doing any of this? I mean, some of these things are not pertain to people in sales, but I will get to the sales part. Then the quality control, I just summed it up here because most of the time, the quality control is lumped with the manufacturing. Sales and marketing practices. This is what concerns most of you here, I guess. So, who are hired for these functions? I'm not really sure how to discuss this, but if you are a marketing person and you are responsible for hiring sales reps, the onus is on you to hire someone who has the flavor. There are some people that are not just good at something, they don't be good at something. So, how robust is the training you got? Is it, how about the initial one and the subsequent training? What is the clinical competence of sales and marketing team? And this is very important because this is the only thing that can make you think whenever you're in front of a physician what the clinical outcome will be. If you are not versatile with all the disease areas and the competing drugs and all of that, you are not able to be a good sales representative because you're a pharmacist, not a microbiologist or something like that. There are certain expectations any doctor you face expects from you. Can Can sales reps and marketing teams interpret clinical, publish clinical information? See this part of it? Can sales rep act as a resource person for a physician? I was a sales rep for 6 years. You walk into a doctor's office, they're asking something that has nothing to do with what you come from. And they do expect you to have a reasonable answer to it. I don't know if you've experienced it before. So I tell you a story about either a patient or some drug or even a competitor's drug. They expect you to have a certain level of knowledge. Now, most of these things you have to train for by the company. It's you that will make an effort to educate yourself on things like if you see things read, if you encounter things, have a knowledge of it. You don't want to be embarrassed that you don't know it, and this is what makes us pharmacists. How do you carry yourself when you are out there working for the company? How do you dress? How do you speak? These are the kind of things that will influence the doctor to make a decision to write your drug and write it properly. At the end of the day, the clinical outcome follows from them. This one, drug importation, distribution and supply chain management. I gave somebody an example the other day with insulin. What happens to expired drugs? Does anyone know? What does expiration mean actually? Anyone? So below that percent is expired, and this is usually put on the drug, but it can also be accelerated by other things. Heat, temperature is mostly humidity, and the dosage form matters too. So you would say percentage, let's say it has an expiry date of 2 years, but it's stored under terrible conditions. So that means it is not what is used to study that drug. The conditions have changed, so it cannot really be the 2 years anymore, but it's degrading. Sometimes what I'm concerned about is not that it's lost potency, but what has that degrading gone to? What is it? You don't know what it is. I tell people who want to take expired drug, there are two sides to it. One is lost potency, right? The second part is what did it turn into? Why they are telling you not to take it already? Because if it degrades to a certain level, that degraded portion of it, whatever the consequent compound is, may be harmful to you at a certain point. So you want to avoid it for those two reasons. So that means that if you should be conscious of the fact of how you store drugs, because the clinical consequence may be very different, or the clinical outcome will be very different. The patient injecting insulin, and you know insulin is very, very small doses can mean a lot. Particularly for patients who have titrated their insulin consumption to a reasonable level. If that insulin is lost 10% potency, that patient will not have a good outcome. Because what is the transit time? A lot of drugs get manufactured outside of this country and shipped here. How long does it take the ship to get here? What kind of things are they stored in? In what conditions are they received? These affect potency of drugs and subsequently the clinical outcome. Does this process guarantee delivery of the dose that is promised? Probably not. There are certain types of medications that are not here yet in Nigeria. Biologics. Not a lot of it, I don't even know it. Biologics are Anybody know what biologics are? When someone describes a drug as a biologic, what does it mean? So, I don't expect you to know because these drugs are not here. But that's the trend now in developed world. Biologics are drugs that are not synthesized in a certain manner using enzymes that put these moieties together. Some of them are small molecules, some of them are bigger molecules. But they are so specific in what they interact with. Think of it as more of an engineered drug for a particular disease, and not just for that disease. You can't say it's engineered for breast cancer. They will be engineered for a specific type of breast cancer, a certain genomic profile. That's how specific treatments have gotten in developed world. So 3 women may have breast cancer, but they are not the same. I worked in oncology for 11 years, so I'm familiar with how different 3 people, 5 people who have colon cancer. It's not the drug that will treat one person that will treat the next person, because the genomics of their cancer is different. And this is something that can be tested for. What does it do for the patient? It's more specific, reduce side effects, you are more likely to get killed, or at least what we call progression-free survival. Maybe if you use ordinary chemotherapy, you may get a year. But these drugs can give you 3 years, 5 years of progression-free survival, which is the best measure, if I may say, of cancer treatment. How long can you live while on this drug? Some are so short, depending on the severity of the cancer. Some are so long, particularly for hematologic cancers, where the patient is cancer-free for almost 10 years. So depending on how good the genomics are, and if the appropriate drugs to treat that. So now, sorry, this is always about. How do you know what? How do you measure clinical outcome? There is a way, a certain established way. Clinical outcomes are measured. It's only by clinical trial. There's no other way you can say a drug does this, or a drug is better than this drug, or a drug is the same thing. They call it like a me too kind of thing. Or it's cheaper, or it does exactly the same. So I tried to put this on this in one number. I tried to review there are about 4 phases of clinical trials. You shouldn't concern yourself with part 1. Phase 1 clinical trial usually is just a moiety testing, whether it has activity. Sometimes it's in vitro. Then that was phase 0. Then phase 1. Phase 1A, sometimes they do phase 1A for some. It's usually trying to see if there is any bit of in vivo activity. Some pharmacokinetics, some pharmacodynamics. It's when you start from phase 2 that you see things like dosing schedule. That's when they try. How many times can you take this in a day? Some once a day. That's when they establish the dosing of the drug. Anybody familiar with this here? Is anyone? You are familiar with it? Okay. Then phase 3 is the clinical trial itself. Most of these things you see that they hand you over at the company that phase 3 trial. That's where you see all this comparison, depending on the type of comparison. Then there's the phase 4. Phase 4 is usually surveillance phase. They are looking for adverse events. Are there other things he's treating that is not what is registered for? there are many potential adverse surveillance and all that. That's this for. This sometimes is a retroactive study. They are just checking through all the different studies and they're trying to see what they can do. But trial design. We are doing this because I want you to grasp outcomes. There's no other way. There is clinical outcome. How do you prove that? So you should be able to say either a competitor sales rep. How do you prove your drug is better than mine? If they refer you to a clinical trial, you can Google it. And I want you to be able to Google a clinical trial and interpret it for yourself. At least at the basic level. You don't have to be heavy. You don't have to go into the statistics about showing you what to do. Okay. So two are the most common ones. There's the parallel study and there's all are randomized. What is randomization? Anybody know? Huh? Yes, but there is also a science to it, because if you toss up the dice, it's a kind of a random thing that will show up, right? So that's the simplest explanation, but there is a science to randomization too, because on top of the randomization, you are also putting patients in different classes. Okay, the drug or no drug, drug, placebo, drug and another drug. So for you to do a credible clinical trial, those two groups have to mirror each other. You understand? The other one is a crossover where you give a group washout period, then they take the other drug. So that's what you see. I don't want to get into the factorial. Those are the most common to see. But note that they are both randomized. There is also blinding. This usually is blinded to the investigator. Look out for that when you are reading. Somebody gives you something and say, I'm going to compared to your own medicine, your own drug. You want to check was the trial blinded? Who was it blinded to? Patient and investigator, patients and people who will be doing like labs or the surrogate outcomes. Because if you're not completely blinded, then there is an element of bias that can come into interpreting it. So having said that, this is not very clear to you, but I will read it out for you. What I was trying to do is say clinical trial is the fundamental for medical practice. It is the only thing that gives you credibility that you can prescribe a particular product. So, it's needed for effective clinical decision making. The publication format, you can see it a bit. When you take a clinical paper, it's going to have certain things. Please forgive me if I'm repeating what you know, but I think every pharmacist should know this. You don't enter somewhere and they give you a clinical paper, you have no idea what it is. Going to see title, author, of course the name of the journal is published, the title, the author. Sometimes you see the abstract. The abstract is just a summary of the trial. The method, results, and conclusion. Now in between any of these there may be other small subtitles. But the most important thing is there are key questions to ask. And those key questions are, does the trial result even mirror what they are? Is it relevant to what they are talking about? Is it valid? Is the result valid enough for you to ask someone to prescribe clinically useful. There are things that are statistically significantly different. Sorry if I mumble that, but two drugs can have statistical significance in terms of different statistics, but clinically they may not be the same. So is it clinically important for you to use that drug? How applicable is that result for the patient the doctor is seeing? You walk in and talk about a drug. For the patient, it's a big doctor does not see this kind of patient, so it's irrelevant to their practice. There are usually experimental studies comparing one intervention with the other. So, but there are certain criteria that is used to measure whether this intervention are valid or not. The randomization I talked about, this is what it does. It takes a certain criteria like age, gender, comorbidities, and a lot of other factors. Comorbidities, has this patient taken any other drug before? What are previous therapies? And all of that. And if you have 100 patients and have to do 50, they have 50 years, they have to match exactly. You know, maybe it's small variations, but the more the variations, the more you end up with what is called confounding factors. That means whatever result you are going to get from that study is compounded by the fact that the patients are not the same. Let's say you take a paper that is for this antihypertensive. When you look at it, there's something wrong because they didn't tell you the comorbidities of these patients. You don't know. So would you really recommend it for a diabetic patient that is hypertensive? You can't know, because the trial you are reading does not say that this was tried in diabetic patients. You get my point? So these are things, and if you put that result out there with some diabetic, some not, and you do randomize them properly, that diabetes becomes a confounding factor for the analysis of the result. I said, promise I wasn't going to go too deep into that, but it's just to explain to you that they may not be like what they see. So there are other considerations when you are looking at the clinical trial. Look at the site. Where was it done? The ethics committee, we have an ethics committee. Now, let me tell you something about that ethics committee. They want to try in a hospital. And he has to go through the ethics committee of that hospital. Because they are the ones to say whether you can conduct the trial or not. The reason being, is it safe to even try this? And who are you trying it on? Is there an ethical issue with you trying this on humans? Is there an ethical issue to do the drug, the medication, and placebo? When it's an innovation or innovative drug, we are denying that placebo the opportunity to get treatment. So these are what the ethical committee looks at, or the ethics committee. Then government regulations. You have to register this with the government, where I come from. Now, there are issues of chance, bias, and treatment effect. Is that treatment effect accidental? Which is very, remember we are rolling a dice, kind of. So it may just be accidental. Are there factors that can affect investigation, patient selection, randomization, inappropriate statistics? Now that is very important. the sample size of the trial can affect the whole result. And a clever investigator may choose a different kind of statistical analysis just because they want a certain result. So be careful when you see like small sample size or very large sample size, and see how they are, whether the statistics they use is appropriate. That I don't want to get into so much because that's a completely different topic. But it's something you can look at. I talked about statistically significant and clinically significant. Two moieties can be statistically the same, but in terms of clinical significance, you may not use them as such, or you may not interpret the result as such. So there are two ways questions we have in the study results using three filters. Validity, clinical importance, and clinical relevance. Are the study methods valid? Randomization, I think I've called it. Was the list concealed? There are some investigators that may go up to the point of concealing this pathogen. You know, sometimes it's not very often you see reputable companies using such investigators. So, how long was the patient followed up, or how long did they take the rest? Some results are better if it's short term. Some are better if it's long term. But you as a pharmacist will determine, is this enough time for an antibiotic treatment or not? If you are following up somebody with hypertension or diabetes and you are trying the medication in that therapeutic segment, how long can you follow this patient? You have to look for it, because that complication will say how long you follow the patient. Sometimes all the patients that are recruited are not and all of that, or people drop out for various reasons, just because of the patient side effect. But there is an analysis called intention to treat. It's a very good analysis for a treatable population. So everybody who registered for that trial will be analyzed. There is a way to kind of project when they drop off or when they don't show up anymore into the analysis. But intention to treat analysis is always the preferred analysis. For all patient analysis to which they are located, sometimes there are sub-analysis in groups. Okay, this is a group of females. You can't toss a male person in there just because you want to. And the study result is very important. The outcome must be sufficient enough for you to say or sufficient enough for you to say it's better than another drug. Was the treatment effect large enough to be clinically relevant? Sometimes these effects are very marginal, but the company will be pushing for its sales. It's not that it's wrong, but you as a pharmacist, you recommend where I work to recommend which one do you take, which one doesn't. I'll give you an example. With prostate cancer, there are different modalities to manage it. One of the two drugs that are used orally to manage metastatic prostate cancer, particularly those who are castrate-resistant ones. Castrate-resistant means in the absence of testosterone, it's still growing. So there are two drugs, just follow me here. One is called Zytiga, the other one is called but the issue is that sometimes doctors will call and ask for which one to give the patient first. Whichever one you choose is going to give that patient maybe about 2 years of progression-free survival. Understand? But there is a sequence to which you give this medicine, you don't get the benefit from the other. Do I give Zytiga first, or do I give Extended at the name of the other medicine? So if you first give Extended, after 2 years and this patient progresses, you won't get much out of Zytiga. But these are things a pharmacist will tell the doctor because you researched it. They ask you which one do I give first. So it's up to you to go and find out, and then you feed them back. But you also have to know how to read this paper, because if you don't, you can't. And whatever conclusions they are making have to be the questions they ask. You can't treat, say, I want to find out this service, but in your conclusion they are concluding something completely different. Or you try to be smart. Sometimes that trying to be smart because they are using, so I told you about surrogate markers. And they say, oh, we reduced the PSA by so. That's not what we said. Is the study population similar to the one you see in your chart? It's reproducible because the whole thing, this is science. It has to be done in such a way that any other clinical setting can reproduce that. What are your patient's personal risks and potential benefits? I will show you later how you use maybe 2 particular numbers when you finish reading this to see whether how that product performs or compared to another one. Of course, we talked about quality of the study, whether the results are valuable. Is it really important enough? I've seen companies promote things that do not, like I said before, marginally just okay. But it's up to you to give that paper to do. You can make out that, okay, what is the hype about? This is just a small thing. We talked about surrogate markers. So, there are certain outcome effects that we use to determine how good the paper is. They are expressed usually 4 different ways. There may be other things. And remember, outcomes are different things. Take an antibiotic. The outcome that's presented is different from if you're taking oncology medicine. So keep that in mind. If you do a test, there's no bacteria anymore, whatever. The patient has no more fever, and all of those are outcomes from an infection. Is for cancer, their overall survival, overall response rate, progression-free survival is the most common one. How long are you? This is cancer-free. There are some people that be dramatic to get a cure, but they don't use that much for evaluation. They use, you are not cured of the cancer, but you can, it's not growing, and it may not grow for 10 years. In fact, sometimes it seems like you are cured. I've seen it, cancer, all manners of cancer. Then 10 years later, the person is in your cause. And the funny thing is that cancer cells can hide for that number of years. The body has just, your body fights cancer every day, even as you are sitting here, you don't have it. Because look at our body and the number of cells. Each one of them has the capacity to mutate at any point in time, and they are mutating. But if you heard of our immune system, like the natural killer cells and all of these immune systems, that they are the ones that kill these cells that have mutated and don't look like your cells anymore. So they are actively being killed. There are some circumstances, like you say, what do you eat or do you smoke? This thing becomes a reduction in your immune system that leads to you not fighting this mutated cells as you should. So that's when you get cancer. This is maybe a simplistic explanation of it. But know that as we our cells do mutate, either by help by environmental factors or just spontaneous mutation. So to express it in what got me into that was I was saying the outcomes. So there is a relative risk. Now that relative risk, the relative risk is the ratio of one versus the other, or the experimental treatment. And that risk is, what he's saying is that if you take drug A, what is the relative risk that you didn't take drug A? So that's how it's expressed as a relative risk. Okay, so that's it. It's not used much, even when you read clinical trials, not very much. The risk difference, or the absolute difference, is that taking the probability of one and subtracting it from the other, and whatever difference you see is the absolute difference between the two. This is one of the most important. I told you I was going to tell you two things that if you look at any clinical paper, look out for those two. And one of them is this risk difference. You subtract drug A, the probability that it will do well, or let's say, let me give you a typical example. 100 people on each drug A and drug B. If 29% of people in drug A were successfully treated, that's 0.29, right? Then 50% in drug B, that's 0.5. So if you take that difference, that's the absolute difference. Now that absolute difference, the reciprocal of it is what is called numbers needed to treat. Now that sounds fancy, but what it is generally the lower this number, the better the drug. What it means is that this is the number of patients you needed to treat to get one more patient treated successfully. So you can see the lower it is, the better the drug. So when you are reading a clinical paper, look out for this two, the absolute difference and the number you get to treat. There's another one called odds ratio. It's like relative risk too. What are the odds? You know, for those people who play things, what are the odds that you gain from this treatment compared to the other one? And it's measurable, and most of these things are calculated. And when you take them, there's something I want to take you a little bit. Before you make that, look out for sample size. There's something called p-value. I don't want to dig into all of that, but think about p-value as level of error that is you are willing to permit for this trial. There's another one called confidence interval. Most of the time you see it at 95% confidence interval. That is just saying that within 95% you are certain that so will happen. And that value needs to be defined ab initio. It's not something an investigator will come later to determine. And some of it, both the p-value and the level of significance is calculated using the sample size. Do you think you can see a clinical paper now and recognize some of it? Some of these things? It's not something you are familiar with. What our advice is, when you get back to your parents, just go to Google, search treatment instead of so, whatever you like. It will pull off things. Now, there are some things I'm going to let you know now. Just because it's in Google, it's even made worse by the AI stuff, does not mean that it's bad. So that's what I want to discuss now about searching for clinical information. You can Google it, but that does not mean it is correct. But now the AI system seems to have gotten a lot better, because it will give you something and give you a reference where you can go and find more information. So other considerations: clinical information search. Before you research, just like I said, define what you're looking for. If you don't know what you're looking for, you end up with a lot of stuff. Where to search? Internet is usually the most common one. Keep in mind the length of time. When was it published? Which journal was it published in? There are journals that are reputable. Let's say the Indian Journal of Medicine, for instance. Is the source credible? Type of study, you have to check that. Is it authors? Is there a sign of bias? Is it being sponsored by a company? It's also a sign of bias. There are certain traps you have to avoid. Like I said, AI is one of them. Try and look at the list you are given and look at more reputable sites to go and search. And if you are searching for a particular topic, pick 2 or 3 of them and see how they compare to each other. So on that note, I will just summarize so that we can you have to appropriately interpret whatever you are reading using those 3 filters. Validity. There are certain exceptions to clinical trial, don't get me wrong. There are cases where, because of where I practice, we call last ditch effort. Just because a patient has tried all manners of medication, let's say cancer, a breast cancer patient, there's something we call last ditch effort, where the drug seemingly not tried in that area, but the doctor has a reasonable conviction that it will help that patient. Like I said, this is after they've tried other things. There are sometimes you see extrapolations. You may not have seen a trial in that disease area for that medication, but due to the activity on certain receptors, a physician is allowed for off-label use. So you cannot deny that dispensing that medication for that patient because it's written off-label. For due diligence, because there in the US we have what is called corresponding responsibility. You can't say, the doctor can't tell you just have to dispense what I wrote. No. The pharmacists have the right to deny dispensing a prescription. So you call the doctor. If they provide evidence that satisfies you about what they're doing, particularly in off-label use, then you can dispense. Sometimes the trial may not match, but it gives you an inkling of what they're trying to do. So you dispense the drug. On that note, I will stop there. I had other things to share, but there is not. So any questions, please? I hope you guys enjoyed it. Sometimes these things get so technical, but as a pharmacist, we have how many here? Any of us should be able to pick up a clinical paper and analyze it. If you just use the criteria, you may not be perfect initially, but you should be able to ask for a publication from anyone, any professional, doctor, pharmacist, anyone, and you can interpret it yourself. And if it's not appropriate, you will also know something is not right in this trial. And there are a lot of shady trials out there because people are trying to market what the drug is not. Any questions? Yes.
My name is Lucina, and I currently work in the QC department. So, however, my long-term goal is around the oncology. So now, for you as someone that is actually in the oncology space for over 10 years now, and I might work in Nigeria, so there are more updates over it. So for someone that is getting started, so now my question is actually out of sales or more in the oncology. So for someone that is an early career researcher in oncology space, what space, what can answer at the moment, and there is a lot of involvement and training. So what are like, so my question now is, what are like the answer for some person to actually get an edge?
You mean in the global fight?
Yeah, like now talking about oncology,
Yes, the R&D, cancer, there's a paradigm shift in cancer treatment. And this is where all the R&D is going now in biologics, in specific moieties that target specific mutations for the cancer patient. That's where they are. So, and the reason is it's more specific for each person. All the research and money in R&D in cancer now is being poured into this area. And it doesn't even matter which type of cancer. They are going to cellular levels, enzyme levels, mutation levels to see the genetics of this cancer. Then they try to develop drugs that can latch onto that specific. Let me tell you something very interesting. Somebody told me that you can eat certain kinds of fruits. Diet can cure, well, they say cure cancer, which is very ridiculous thing to say. I don't know who you are. But diet can help you fight cancer. If you are a cancer patient, cancer cells do not have the capacity to use ketones as an energy source. So if you can get yourself to ketosis, it will not kill the cancer cell. That's if you leave sugar and you're eating certain diets, which is possible, and it gets you to ketosis. Your body, normal cells can use ketones. It's a source of energy. A lot of athletes do that. Then the cancer cell is being starved. There's all this story about diet and cancer. I'm just telling you what it is. Get to ketosis. Cancer cells have been found not to be very good at using ketone as source of energy. So they want sugar. So get into ketosis. What it does is that it doesn't kill the cell, but it makes them weak enough to respond to treatment. That is, so sometimes you see a multidisciplinary, bring the dietitian in, bring the physical therapist in case they have a debilitating. So you have teams, certain individuals around this patient to advise on diet and all that. So they think that is a more holistic treatment of the patient. So this is just by design. But the R&D is now focused on more targeted therapy than anything else. I hope that answers the question. Ladies and gentlemen, thank you for listening. If you have any, I can send this to your people if you want to. Yeah, I will send it to them if you want it.
We need it, sir. We need to send it to them.
Yeah.
They promise they'll send everything to
okay. I'm sure they will ask. All right. So I'm around. I'm retired. So if If you want to, I'm still around till tomorrow. If you have any questions, you can ask. I retired and relocated.
Thank you.